Okay, lecture 6, we'll talk about some of the antibody, which is same as immunoglobulin based detection assay, protein detection assays, such as Western blot and ELISA. And then we'll also talk about some other assays that allow the detection of protein-to-protein interactions inside of cell or outside of cell. Okay, first, antibodies, immunoglobulins. What are antibodies? So antibodies are huge complex proteins. Many of them are glycosylated that are made by immune cells, right? B cells, okay, in your immune system, plasma cells. And then they are made up with 4 different polypeptide. So they have a quaternary structure. When you say quaternary structure, what does that mean? A protein is made up with more than single units, subunits. So that's what quaternary structure is. So if you look at a single antibody, it has the 2 large polypeptide and 2 small polypeptides. So it's typically made up with 4 different polypeptides. So 2 of them are called heavy chains, large heavy chains, and the other 2 are called small light chains. So your antibody is made up with 4 different polypeptides. Okay, and what does it do in your immune system? It is involved in antigen recognition or antigen binding. Typically those are antigens. Those antigens are part of the pathogens that are coming into your immune system. So maybe a surface protein of viruses, or surface proteins of bacterial cells, they recognize and bind. So this is a typically antigen binding site. Okay, specialized in antigen binding site, because they have very unique special structures. Okay, so then do we have only one antibody that recognizes one antigen? That's not true. We have billions of antibodies in our immune system that have all different structures here and binding affinities here. So that's how we can recognize so many different pathogens that can come into our body system. Okay, so that's their main function, recognizing antigens on the pathogens, infectious agents, and then they activate the immune cells. So immune cells will fight against those antibodies. So antibodies are kind of defensive mechanism. Okay. So antibodies or immunoglobulins are protein molecules produced by your immune cells, lymphocytes. Mostly immune system are only existing among the mammals, right? And they protect organism by binding to, like I said, the foreign molecules that are called antigens. And then they activate the immune system to fight up. So antibody here, right? And neutrophil has antibody. Another antibody here, eosinophil, okay? And there's another antibody here, macrophages. And there's another antibody there. To read, there's another immune cell there, okay? Natural killer cells, at the NK cell, that's what it means. And if it's a target pathogenic cell or virus, they can bind and attack those pathogens. So antibodies can bind on these, and they can be on the surface of these immune cells. Antibodies have generally Y-shaped structure, so Y-shaped, right, made up with 4 polypeptide, like I said. And if you can designate some parts of the antibody, we call this as antigen binding site. So we call the FAB site. F means it's a fragment of antigen binding fragment. That's what FAB is. That's his antigen binding fragment. So they have a three-dimensional structure that fits on the antigen structure. Yeah, that's the main function of the antibody, Fab region. And this is FC region. C means complementary. So the sequence of this area is very same among the antibodies that belong to the same family. Same family antibodies have the same protein sequence in this FC portion. So FC portion is normally involved in membrane binding. So immune cells membrane, they bind there. Okay. So each antibody, like I said, has 4 polypeptide, 2 identical light chains, and 2 identical heavy chains. Okay, and then each polypeptide has a constant region, FC region, and the variable region where that FAB region is located. Okay, so each antibody, single antibody, can bind 2 antigen molecules because there are 2 FAB Okay, antigen binding regions, 2 antibodies. Okay, antibodies classified based on the 5 amino acid sequences in the C region, okay, of the heavy chain. So based on the sequence difference, there are immunoglobulin A family, D family, E family, G family, and M family. Okay, so 5 different antibody families exist there in the mammalian system. Then how do you make antibody? So of course your immune cells will make them inside of the cells, right? But for your research, for your purification, you need to make antibodies for your purpose. So then can we make them somehow? The answer is of course yes. You can make your antibodies and use them for your research. Okay, when you do that, there are 2 different types of antibody molecules that you can make. One is called polyclonal antibodies, and the other one is called monoclonal antibodies. Polyclonal antibodies and monoclonal antibodies. So what's the difference? The difference is the way they are made. So polyclonal antibodies is basically made by animals, mammals. So you can use a mouse or rabbit or goat, any kind of lab animals, if you can grow them pure in good condition, by injecting antigen, the proteins you want to have your antibody bind. Okay, the purified protein can be injected into these animals, like people getting vaccine shots. Same principle is here applied here. So the animal is injected with an antigen, okay, once, twice, maybe 3 times, maybe about a month period, and then inside of this animal's immune system, yes, the immune system is activated and there will be a bunch of antibody made together. Okay, so that's the basically polyclonal antibody. After a certain period of shots, antigen shots, you can bleed the animal to get serums out, and serum will have a bunch of antibodies against your antigenic protein. So if you isolate those mixture of proteins, we call those polyclonal antibodies. Basically, that's simple way of making antibodies. Okay, so polyclonal antibody. Why is that polyclonal then? Because it's not just single antibody that will recognize your antigenic protein. There may be multiple antibodies that are being made by different immune cells, and it's a mixture of those antibodies together. It's called polyclonal antibody. Okay, makes sense? So maybe a rabbit is injected by your antigenic protein, okay, multiple times. After that, you get the blood out and spin it down, get rid of the blood cells, and the rest of it is serum. The serum is antibody-rich source, so that is how you make polyclonal antibodies. Okay, very simple, straightforward. Then what is the monoclonal antibody? Okay, the difference is, of course, monoclonal means what? There's only one antibody in your antibody mixture. Polyclonal has maybe multiple antibodies that are mixed up together. That's what polyclonal antibody is. But monoclonal, your antibodies are made up with single antibody. That's what monoclonal is. Then how do you make a mixture of just single antibody? So the process of making single antibody is more complicated, of course. The way it's making is, so you have to start out with an animal, of course. In this case, maybe a mouse. Okay, lab mouse will be injected with your antigenic protein. The starting method is the same. A mouse is injected with your antigenic protein, but after that, okay, that mouse will be sacrificed, and from its spleen tissue, mouse spleen tissue, you will collect some B cells from mouse spleen tissue. So that's where a bunch of antibody making cells are located. So you'll collect a bunch of those B cells from the sacrificed mouse who had those series of antigen shots. Okay, and then those B cells, okay, antibody producing cells, will be fused with another B cell. So this B cell is cancerous B cell, called a myeloma cell. Myeloma is a blood cancer cell. So it's a B cell that became cancerous cells in myeloma. So if you make a fusion of these two cells, B cells coming from sacrificed mouse and then cancerous B cell, okay, we call that fusion cell as a hybridoma. So you create bunch of these hybridoma cells between the B cells and myeloma B cells, and you screen those hybridoma cells, okay, with the antibody producing ability. Okay, you may wonder now, why do you have to fuse these two cells? The reason is B cells will give you the antibody you want, right? So B cell is a antibody producing cell. There's that ability. But myeloma cell is a cancer cell. And what benefit does it provide? Cancer cell can live forever inside of a test tube, right? That's the characteristics of cancer cell. By fusing B cell with the cancer cell, now you have a hybridoma that can not only give you antibodies, they can also live continuously inside of a test tube. So you can become a factory that will give you antibody continuously, right? That's why you have to make a hybridoma cell. Okay, so among multiple hybridoma cells, you pick one hybridoma cell line that will give you the best antibody, and you culture that cell continuously, and from those cells you will be able to make one single antibody. That's a monoclonal antibody. Okay, so that's how it's done. Again, you have a mouse maybe, okay, and inject mouse with your antigenic protein, okay, about a month or two, and then this mouse is self-sacrificed and collects some B cells, multiple B cells, and fuse those B cells with myeloma, the cancerous B cells. So when you do that, those two cells together become hybridoma cells. By growing them in a selective medium, you can separate out hybridoma cells from non-hybridoma cells, okay, and collect those hybridoma cells, kind of separate one cell at a time, and from those multiple hybridoma cells, choose the cell hybridoma that gives the best antibody and culture that hybridoma cells continuously. That's going to be the antibody producing cell. If you collect the antibody from those cells, that will be the monoclonal antibody. Okay, that's how you make it. Same process. B cell, myeloma cell, fuse together hybridoma cells, and pick the best hybridoma cell and collect antibody from there. That's the monoclonal antibody. Okay. So if you have those either polyclonal or monoclonal antibody, then you can use those antibody molecules for your analysis, maybe some protein detection method, right? One example is called Western blot analysis. It's kind of old-fashioned, detection method, but the principle is again important, and that principle is used in the process like ELISA. So we'll talk about that first. So Western blot analysis, we talked about Southern blot analysis, Northern blot analysis, just like that. Western blot analysis is a detection method, but this is not DNA or RNA detection method. Instead, this is a detection method used to detect a specific single protein. That's what Western blot analysis is. Okay, now how does it work? The process is similar. You have to separate out your target protein molecules using electrophoresis. In this case, what SS page is the electrophoresis you will have to use. Once that's done, those proteins on the SS page gel will be transferred from the gel to a membrane. Okay, once that transfer is done, you will screen that membrane using antibody. Okay, and to find the protein that you are looking for. That's the Western blot analysis, basically. Okay, so as this page, okay, and then you transfer to membrane, nitrocellulose membrane or PVDF or whatever the membrane you use. That's fine. And then after transfer is done, you will have to first block the non-specific binding site by doing what? Pre-hybridization. It's the same like Southern and Western blot. Southern and Northern blot. Okay, once that blocking is done, then you will add your antibody to find the protein that you're looking for. Okay, and once that's done, you will have to detect where your antibodies are sitting. Okay, for that you have to use a secondary antibody, which is the antibody that detects your primary antibody. Okay, and the secondary antibody has a special function because secondary antibody is normally conjugated with an enzyme. Okay, and that enzyme can break down the substrate into a product. That product will give you some kind of a signal, maybe color or light. So that's how you can detect your protein that's binding with the antibody, two different antibodies. That's how Western blot works. Okay, so I'm going to start right here.